Okay, great. So now I'm going to introduce our second speaker. So our second speaker is Shreyoshi Sur. She's a PhD candidate in the chemistry department at the University of Rochester. And today she's going to be talking about molecular dynamic simulations.
Hi. So first, I wanted to talk a little bit before going into my technique that our lab uses. I wanted to talk a little bit about some of the biological systems which I'm going to refer to throughout my presentation. So as Chandani talked about, cells are basically all living beings have cells, and they are the basis of life. And then inside the cells, there is this nucleus in which we have all the hereditary information. The cells are encapsulated in a cell membrane, which basically separates the cells from its environment, and it can maintain, basically if there was no cell membrane, water can gush into our cells and burst them open. So basically the cell membrane protects our cells. The cell membranes have what I call the water-loving part, so a hydrophilic part at the top, which interacts with water, and then inside the cell membrane are these lipids, which are long hydrocarbons, which doesn't like water. So it separates, it kind of acts as a barrier between the inside of the cell to the outside of the cell. One of the main constituents of cells is proteins. They come in various shapes and structure. And some of the functions that protein can perform are like it can act as a catalyst in different biological reactions. It can help the cell communicate with other cells. It can also provide structure and integrity to the cells. So proteins are really important. Some of these short, small proteins are called peptides, and proteins are made of amino acids, which are organic molecules, small organic molecules, and they come in different, so the amino acids combine to form long polymers. So this is basically each amino acid of different kind, and then they form these long polymers of protein, which is called the primary structure of the protein. And then the protein can come into different shapes, and that's called the secondary structure. So this is our secondary structure of a protein. And then proteins have different shapes. So they can be either beta sheet, which are these sheet-like structures, or they can have these helices, which is called the alpha helix. So these two proteins are actually one is from silk fibril, and the other is spider web, both of which are very thin. So if you look at macroscopically, they just are threads. But microscopically, they have such different structures. So I wanted to say that before I go on. So the other thing is that these proteins can also be inside the membranes. So when they are inside the membranes, they have a specific shape. But if you want to study them in different spectroscopic techniques, we have to extract the protein out of its environment and then look at it. So, but that's hard to do if they are inside the membrane, because once it's outside, it's in water, so it is not in a lipid environment, which it was there in its original state. So that's why we, and in all these techniques, has the same thing. We have to extract the protein out before you can study it. So that's why we use molecular dynamic simulations, which are computational models in which we can look at the proteins through your computers, basically. So, that's these also the biological molecules are very dynamic. They are ever changing, and to study them using one spectroscopic technique is hard. So the molecular dynamics provides us a tool to study these ever dynamic molecules in our computers. It's also, we can also use MD simulations to calculate some of the experimentally observed properties. We can also use it, since we are building the system of the of the protein, we already have more control of the system. What are the things that we want to add in it? Whether we want water in it, whether we want lipids in it, we have more control over it when we use molecular dynamic simulations. So I made a small workflow to look through each part of the technique. So for MD simulations, since we are doing it in computers, we all need first the coordinates for all the atoms of the protein. Then if we have all the coordinates for the atoms, we also need what are the forces that are acting on these atoms. If we know the forces, then we can determine where is, we can put it into different equations of motion. Yeah, we can use different equations of motion to know where is the atom going in the next step. And if we know where it's going in the next step after a small amount of time, so in our case, we use a very small amount of time, so femtoseconds of time to see how a single atom is moving. And then we can know where the next position of these atoms are. And we keep iterating this to get a trajectory. So that's how a simulation looks like. So they keep moving. And I'll go through each part of this workflow. So the first part is the coordinates for atoms. Where do we get them from? For proteins, there is this useful tool called the protein data bank, where we have a lot of the structures of the protein with each of the coordinates of the protein in them. So these are derived from extracrystallography or cryo-EM, which are like modern spectroscopic techniques to study the proteins. And then we can look at what are the forces acting on these proteins. So proteins, as I said before, are made up of amino acids, which are organic molecules. They have covalent bonds between them, and the organic molecules have specific bond lengths which can be between them. So carbon-carbon will have a specific bond. If they're single bond, then there's a specific distance between them. So that's our bonded terms. Then again, different carbons will have different angles between them, and they need to have that angle to define how in real systems they are staying. So that's our angle term. So these can be 3 different carbon atoms at specific angles, and there's a force acting on these atoms to keep them in that angle. And then there is the dihedral term. So if there are 4 different atoms connected, how is that in 3D? How are the angles between them affecting the structure of these 4 atoms? So then if we have all the bonded terms, there is also electrostatics. So the atoms can be charged, and so they have electrostatic charges between them, so electrostatic force acting on them. And we define them using the Coulomb's law, which is inversely proportional to the distance between these atoms. But these are usually non-bonded terms, so these atoms can be non-bonded and far away from each other, but due to the structure of the protein, they can be close to each other. So that's our Coulombic force acting on them. Then there is also weak intermolecular force, which keeps the protein or whatever biological molecule of interest in shape so that they don't collapse into one big blob, basically. So these are the things that are the forces acting on the atoms. So we have the coordinates of the atoms from protein data bank. We have the forces from these data of bonds, angles that we already know. Then we can use Newton's equations of motion to see how does the forces affect the initial coordinates of the atoms. So we know that the forces, and we know the masses of each of the atoms because we know whether they are carbon, hydrogen, oxygen, and then we can get the acceleration from the forces. And then we also assign initial velocities to these atoms when we get them from the protein data bank. So we have the coordinates, and we randomly distribute the initial velocities on each of the atoms. And then we can use these, we can put them in the Newton's equations of motion to get new velocities and new position. So after a small amount of time, we'll know where did the atoms go and what are the new velocities of the atoms. And we repeat this process iteratively So we have the atoms, we put the forces, and then they move, and we repeat this process iteratively to get the trajectory of the atom. So this is mostly the basis of MD simulation, but we can also control the pressure and temperature of the systems. Some of the limitations of this process is that to get all these, to get all the data, run the small equations of motion on hundreds of atoms, it takes a lot of computational time. So to get like a microsecond of data, we take months to get them. So that means that we are spending a lot of time getting a very small amount of data, and we want to compare this with experiments which can run for minutes or hours. So what we can do is there are other ways where we can accelerate this process, get more data. So to do that, what we do is we can coarse grain the system, where if there are 100 atoms making up a protein, we can compact 4 atoms into one particle and then run it. So that way we are reducing the number of atoms in the system, but we are increasing the process of how much time we can get for how much time the simulation can run. We can also use explicit and implicit solvents. So instead of using all the waters and all the lipids, we can create it like a force acting on the atoms of interest. So if you have a protein in a lipid and we have the lipids as implicit solvent, we are just acting some forces on it instead of defining each atom in the lipid, each atom in the water. So that's also a way of enhancing how we can increase the time in which we are running it. So I wanted to show some of the softwares and force fields that we usually use. So these are the softwares that we use in our lab. They are readily available. They are open source, so you can just download them, and you can grab a protein from Protein Data Bank and run them in these softwares. And then you have these are the force fields that we use. So the forces are I talked about, the covalent bonds, the angles, and even the electrostatic charge, van der Waals. They can all be found in these different force fields, and depending on how coarse grain we want the system to be, so how we want it to be, whether we want a high definition all atom system or a low definition coarse grain system, we can choose which force field we want to use, which are also free. So you can grab all these and run them in your desktop. So our lab actually works on membrane proteins. So I wanted to tell you about some of the things that we can calculate using MD simulations of these proteins. So as Chandani mentioned, some of the things that you can do with a protein is you tag parts of the protein and see how much distance is between them. So if they are close to each other, you can calculate the distance between them and get the distance data over time. But when we have MD simulations, since we know the coordinates of these atoms, it's easy to calculate the distance between these coordinates. And we can also we can get the same data that we get from single molecule faster, but we can compare them and also get more information. So instead of getting just one distance, we can get multiple distances and see at a different part of the protein what's happening when we do MD simulations. But there is one thing that we should keep in mind when we are running MD simulations, that the time. We need to have it go through a longer amount of time so that the system has the opportunity to go through all the possible coordinates that it can go through. So I wanted to show some of the applications that we do in our lab. One of the things is antimicrobial peptides. So these peptides are actually present in our bodies, on our skin, which act as a defense mechanism. So these are short peptides which protects our skin against harmful pathogens. And how they act is they damage cell membranes. And this protein that I work with specifically attacks fungal cells. So it has a very specific selectivity in which it attacks. And it has this is the peptide part which loves water, and this is a carbon chain attached to this peptide part which hates water. So when these proteins are released by the organism it is in water. So the water-loving part forms an outer covering, and the water-hating part resides inside, tightly bound. And this is called a micelle. It's what soaps do when you put them in water. So these are these micelles present in the water, and then we can use that to see how they will interact with the membrane. Since I said before that they damage cell membranes, so we wanted to study how does these micelles damage them, and if we change the lipids present in the membrane, how would that affect the binding? So I have a short movie to show you guys about how they bind. So if you see the micelle goes, it kind of hovers here, and then it will hover for some time, and then it will plop down. So that's how it binds to the cell membrane. We wanted to see, even after binding, if these peptides bind, how does that affect the cell membrane? So if this is a side view, so we are looking at water here. Water is not there, but there is explicit water outside, and the micelle is interacting with that. And then this is the lipid part. So this is the phosphates, top part of the water-loving part of the membrane, and the inside part of the membrane. And then we flip it. So now we are looking at the top part of it. So we are looking from the top on the cell membrane. This is again the big micelle that you guys saw bound to the membrane. I want you to see how does this micelle disintegrates over time. So I'll play the movie, and you keep looking at it. It's basically the small parts of the peptide. Since it's an aggregate, it keeps going away slowly, and then it will slowly disintegrate into single monomers. So that's how it interacts with the membrane. It kind of binds, and then it disintegrates. So it's harder for an experiment to look at this, because this happens very fast, and since it's difficult to isolate these proteins from its environment, it's harder for it to understand how they're affecting the cell membrane. We also look at synuclein. So this is a protein which is found in the brain cells of Parkinson's patients, and these form these fibrils, which are very toxic to the brain cell. My lab looks at how does one of these peptides bind to the big fibril. And these are different shapes of the same protein. So this is the top view, this is the side view, and these are the shapes that I showed before, the secondary structures. So these are beta sheets structure of it. And I'll show, if you keep looking, this is the monomer, and it's trying to bind to this fibril. We wanted to look at, is there any specific way in which it binds? So we look at the monomer hovering around, then it swiftly binds. So it's hovering, it finds a specific interaction, and it boops in. So we wanted to also look at another protein that our lab works with is rhodopsin, which is responsible for vision. So it's present in our eyes, and it detects light emitted, reflected in our eye. So it's responsible for vision, and it's embedded in the cell membrane of rod cells. So this protein is actually inside the membrane, and these helices are embedded inside the membrane. And this protein goes through a lot of shapes as light is emitted on it. My lab looked at a specific state, which is called the dark state of the protein. And we wanted to see this particular part of the protein. I'm sorry. So we are interested in a particular loop of the protein, which is this part of the protein, and we wanted to see how the interactions in this part gets affected when the lipids of the membrane interacts with the protein. So here is a movie, and I want you guys to look at this part of the loop very carefully, because the change happens very fast. So the protein is interacting with the lipid, and these two amino acids, one is part of this helix, and the other is part of this loop. And they are both charged, so they are interacting electrostatically. And we wanted to look at, as ligand binds to this protein, how does this loop changes? So if you look carefully at the loop, it will change very fast. Keep looking. So as this part is moving, yeah, so now these two have moved apart, and the loop part, which was a loop before, now becomes a helix. And this is important because this shows that the ligand, that the lipids in the membrane is actually acting as a ligand for the protein to change its shape. So this was something that our lab studied, which is hard to do when you're looking at through experiments because it's such a minute detail. But it's important because we want to know how this protein is responsible for vision, so we want to know all the states it goes through. There are other applications of molecular dynamics. We can look at drug binding, since a lot of the proteins are responsible for diseases. This is a very, there are small organic molecules that can bind to the proteins and that can act as potential drug candidate. We can also look at metal surfaces using MD simulations. We can look at RNA and DNA conformational changes using MD simulations. All of these require different kind of force fields. So depending on the molecule of interest, your force field will change. And then also nowadays, a lot of labs in University of Illinois, Urbana-Champaign, they look at actually full cell simulations, like the whole cell. They are simulating the whole cell and looking at millions of atoms interacting with each other. So I would like to thank my lab and the T32 grant, which gave me this opportunity, and the University of Rochester. Thank you. Yeah.
If you go back to like the alpha-synuclein slide.
I'm sorry, one sec. Yeah.
So does the simulation with that one, does that allow you to like compare how like different environmental products would like encourage that kind of like the dityrosine cross-linking to form those fibrils, or is it just like?
So these are not actually connected. The fibrils are basically they are aggregating. So they're not really, they don't have any covalent bonds between them. Yeah.
So when they aggregate though, like if I remember correctly, don't they form like dityrosine cross-links, and then like certain metal ions are like implicated in like encouraging those to form?
Yes. So the thing is, the one that I look at, it's a small part of alpha-synuclein. They do interact with metals, but we are trying to look at how does one monomer binds with the protein, not with the metal. So how does it interact with the protein part of the fibril?